Oh, you have
No, you sit down. I'll sit right there.
you should go ahead and get the card in here. I want to welcome you all to our first Lunch and Learn series this year, funded through a HRSA grant. As many of you know, we have been funded through Health Resources Service Administration for the last now 7 years, I guess. We have 2 more years worth of funding. David Ledbetter was the PI the first cycle, and the second cycle we have I've continued to be the PI for the grant, and we have tried to build the infrastructure for the genetic gaps in the Southeast region. That was the main purpose of this grant. And there are 7 regions, and we are called the Region 3, or Southeast Regional Collaborative. And we also have a website now to update all the activities, which Adria has wonderfully worked on. So I would encourage you to stay to go visit that website once in a while and see what the activities are, and hopefully we'll keep you posted. But for today, this is first of our Lunch and Learn series, and it will be taped. We have Louisiana, Alabama, and Florida who are going to be joining us through the RT1 system here, and we'll be taping this lecture too. And most of you might have already met, but if not, Brian Pike is the project director for the HRSA grant, and I'm going to ask him to come and introduce our speaker.
Thank you, Ronnie. First, I'd like to give a very warm welcome to Tim. He drove all the way from South Carolina this morning. Like I said, probably before graduating. So join me in welcoming. Join me in welcoming Tim. So when Tim and I first discussed the possibility of him coming to speak today, I gave him the overarching topic of challenges and opportunities in molecular testing. Today, he's going to talk to you about some case examples from South Carolina, specifically related to VLCAD and GA1. So Tim is board-certified biochemical molecular geneticist. He's currently the director of the biochemical laboratory in the Greenwood Genetics Center. He did his PhD at the University of Alabama, Birmingham. So with that, I'll just let Tim start.
Well, I really appreciate all of you guys taking time out to come and have a free lunch. But I appreciate you coming and listening to what the work that we're doing in South Carolina. I don't expect it to be something that blows you away or answers 10 questions for you, but I hope it leads to a discussion. And as Ronnie said, I'd really like it to lead to further collaboration between Georgia, South Carolina, ideas about how we can improve newborn screening testing. Basically, what I'm going to tell you a little bit about how South Carolina works, just so you can sort of understand the process that we go through, which is a little different than what's here in Georgia. And then I'll present basically 3 different cases of low excreter GA1s that we've identified, and then a couple of cases of VLCAD, and how we're trying to deal with that in South Carolina. What I'm really interested in is what's going on in Georgia, what's going on in other states, and how we can all learn from each other, or that I can learn from you to give better patient care. When I envisioned what I was going to be doing today, I pictured myself in a small conference room downstairs, and probably the only people that were going to come were the people that had to come. And then this word pizza came in and just sort of blew the whole thing out the window. I got to be real careful when somebody asks me to give a talk that I keep, the limit of participants at a certain. Well, I'll speak to 10 people, but that's the maximum. So when I was thinking about how to do this, I didn't know what sort of level to prepare it in. If any of this is too low. We'll pick it up in a minute. But a lot of people aren't familiar with how biochemical genetics works, or how sort of the idea behind what we're doing. Keep in mind that we were doing biochemical genetics and looking at biochemical disorders before we knew the structure of DNA, before we knew the sequence. There was actually a time before you could do PCR, before there were arrays and all that kind of stuff. And biochemical genetics has a long history in that. And the idea is that there's an enzyme, and enzyme is coded for by a gene somewhere in the genome. And the lack of that enzyme perturbs some metabolic pathway, and you get a substrate that becomes elevated. And this substrate elevation is specific usually for at least a pathway or a group of disorders, or sometimes even a specific condition. The importance of that for what I'm going to talk about today is while that elevation may lead to some sort of pathology, we learn very quickly in biochemical genetics that by understanding what this is, we're already well on our way to treatment and well on our way to potentially alleviating some of the pathology that's in these conditions. And there's a time in a lot of these conditions where even though this compound's elevated, pathology hasn't occurred yet. And that's the true beauty of newborn screening, is that we can get in there. If we identify these patients early enough, we can put them on appropriate treatments, put them on appropriate drugs, and rather than having pretty severe consequences, we can have children that at least have a reduced impact of their disease. And so some of the things we're going to talk about today, especially I'm going to talk about organic aciduria. In case you don't know what that is, organic acids are intermediates in many cellular pathways, including those of amino acids. And so when an organic acid builds up, it can cause all different kinds of problems, leading to changes in blood pH, inhibiting other pathways. A single defect can reach multiple different biochemical pathways. And then we're going to talk about problems in fatty acid oxidation, and that's the breakdown of fats to make ketone, which is really important when glucose is low for the heart and brain. The intermediates of that pathway is something called an acylcarnitine, and the analysis of acylcarnitines has really been the driving force behind expanding newborn screening. And there is a link between elevations in acylcarnitine and organic aciduria. So by looking at acylcarnitines, we can get an idea if there's a problem with an organic acid. So I always put this slide up, especially when I come to big cities like this, just to tell you a little bit about where I come from. And this is the Greenwood Genetics Center. It's in a little town called Greenwood, started in 1974 by a physician who was training at Johns Hopkins and wanted to bring genetics back to South Carolina. Why did it end up in a town of 40,000 people, at the time probably 10,000 people? And it was because there was a family who was working in textiles that was really interested in genetics and providing genetic services. And so my boss did what a lot of bosses do, they follow the money. And that's why the Greenwood Genetics Center is in Greenwood, through a tremendous amount of financial support from one of the main families in town. So you can see we don't have problems with parking. We don't have problems with commutes. This is about 125 acres that sits out here. I don't know what the cost in downtown Atlanta would be of that amount of land, but it's probably a bigger number than there are cells in your body. And then the latest thing at Greenwood is this Center for the South Carolina Center for the Treatment of Genetic Disorders, which is all new lab space and all new clinic space. And so again, I tell you this because you can see Greenwood has a real emphasis in the treatment, and the metabolic treatment program is a big part of that. So Greenwood is here. This is South Carolina. This actually is a screenshot from Southeastern Genetics website. So again, one more reason to go look. And you can find clinics there. We have a laboratory directory of genetic services. Lots of good things on this website, and I agree with the previous comments that you guys should take a look at it when you're having a glass of wine or you're having a coffee in the morning. Hopefully you're not having wine at work, but to each his own. Now most of the newborn screening in South Carolina is done here in Columbia, and then that information gets farmed out to the metabolic geneticists and the clinicians in the state. So we work like a lot of states do. This is going to be nothing new. All the analytes that are measured as part of newborn screening are measured at the state laboratory in Columbia. And then there are three main biochemical geneticists that get to call, as well as the pediatrician, about the positive newborn screen. Most of the follow-up testing goes through the Greenwood Genetics Center. We have contracts with most of the hospitals in South Carolina, and of course our physicians are going to push our services. Well, that's fair or not. It certainly doesn't mean that our work is any better than anybody else's, but honestly I think they do this because they have my home phone number, they have the text home phone number, they can get people there at whatever time they need to, and you can't do that at Quest or Labcorp or somewhere like that. And just as my disclaimer, because you're always supposed to do disclaimers, GGC does receive a handsome sum from the state to fund all this work, and so one of our reasons to bring on more testing, of course, is to justify getting more dollars from the state for newborn screening. So expanded newborn screening, and again, I'm not sure where everyone's at with understanding expanded newborn screening, but what happened is in the early part of, say, 2002, 2003, a lot of states expanded and added what's called an ASO quarantine analysis to their standard methodology. And that allowed them to bring on a huge number of disorders, including organic acidureas and defects in fatty acid metabolism, as well as bringing things like biotinidase C if there were ancillary things all brought in as well. And at this time, when we talk about expanded newborn screening, we're going from 5 or 6 conditions to well over 30. And just to give you an idea, South Carolina has about 60,000 births, which is probably the number of births at one of your hospitals in Atlanta. So we're an order of magnitude different than what goes on in Georgia. And these are the relative small numbers of disorders. We get about 10 to 15, maybe even up to 20 diagnoses a year made through newborn screening. This all comes from Kathy Tomashitis, who's the follow-up coordinator. So one of the problems, I don't want to say problems, but one of the things this presents to us as one of the main follow-up labs for the state is how are we going to help make these diagnoses? How are we going to provide treatment for these conditions? And how are we going to monitor that treatment? And that's really when my former boss came to me and he said, oh, by the way, we're going to need to get involved in expanded newborn screening. He had no idea the amount of time that was going to take for us as a diagnostic lab. Or maybe he did, and that's why he's now retired. So we do follow-up very similar to what a lot of states do. For many conditions, there's a second newborn screening card that's given, and that's used to either confirm the initial diagnosis or determine if the initial screening is a false positive, which is going to happen in this kind of work. Alternatively, the initial screen may be just so obviously consistent with the condition, and so the diagnosis is pretty straightforward. And I give you all of this as background because I'm going to talk to you about 2 problem children that there are in the area of newborn screening, and that's this disorder called VLCAD, and then this other disorder called glutaric aciduria type 2. And I'm so glad to see a couple of people in the audience nod their heads, because I thought, maybe you guys had this all filled out and it just hadn't figured out it hadn't trickled over to South Carolina yet. And what I want to talk to you about was sort of the diagnostic odyssey we've been through, especially with GA1 and 3 low excretors that we've been dealing with. One diagnostic test that I'm going to show you our data on, and I'm very interested in what you guys' data is and how you deal with these cases. And then we'll do the same sort of thing for this disorder, VLCAD. And in essence, what I'm going to tell you is that I don't think these problems are going to go away, but I think we have very integrated molecular and biochemical testing, as well as moving our knowledge into the area of bioinformatics, that we can make much quicker diagnoses, and we can, to put it frankly, scare a lot less parents, because that's one of the things we hear over and over again. Yes, you made 15 diagnoses, but you scared 300 people. And I remember when my daughter was born, I was scared enough, let alone having somebody come talk to me about what I'm about to show you. In fact, to this day, I'm still mad at the nurse. The slit in her foot when they did the newborn screen. I mean, it was a little much. Did not need to do that to my newborn child. I was going to write a letter. And I knew what was going on. But I was sleep deprived and scared. Okay, so GA1 stands for glutaric acidemia or aciduria type 1. And the prevalence that I was given was about 1 in 100,000, which actually, now that I think about it, our data supports. These children are going to have normal development and no other clinical symptoms at birth. And then usually they'll have some sort of acute encephalopathic episode that's secondary to, or that is followed after infection or fever or something like that. And when that happens, they'll have profound hypotonia, loss of motor skills, some of the other things we list here, and they just don't recover from it. And so the way I think about this condition, and I'm not a clinician, I'm a lab guy, is that you sort of get one shot at it. You can prevent this, and once they have one of these episodes, maybe they can recover, but there's a good chance they won't. And so you certainly don't want to miss them. There might be other disorders that we could think of where, if your diagnosis is delayed a year, and I'll actually give you an account of that. There was a case in South Carolina of a patient with MSUD that screened negative, that screened normal, and then at about a year of age presented to a neurologist. But luckily she had sort of an ameliorated phenotype, which is still, if that's a relative word, I think we're all aware of that. She died at 2 weeks of age. But she is under treatment and she is improving, and I'm not sure once these kids have their episodes, they're going to do that. So what you're looking for on your acylcarnitine profile, whether it's newborn screening or diagnostic testing, is an elevation of a single compound called gluteral carnitine. Our designation for that is C5BC. So that's the term I'll use to describe gluteral carnitine. And that's really in the absence of other elevated acylcarnitines on the profile. What usually happens after a positive gluteral carnitine test is you do follow-up plasma acylcarnitines. You look for elevated C5DC. You look for carnitine deficiency, which would prevent this elevation from occurring. You look in urine organic acids for these two guys. I'm going to talk to you about doing urine acylcarnitines. And the problem with both of the conditions that I'm going to talk to you about is that you can be fully affected and have relatively small elevations on your blood screening. So when you got to make this diagnosis, if you don't have real straightforward metabolic elevations, you might have to go to enzyme testing, which could involve a skin biopsy. Again, you get a scared parent like me. You're going to do what to my kid? You're going to take a little hunk of skin? Yeah, let's go, buddy, right now. Or you got to do DNA sequencing, which of course is less invasive. But again, if you try to stick a little kid, and for those lab people in the group, right, they never get you enough blood, and it just goes from there. So again, the problem with glutaric aciduria or acidemia type 1 is there are documented cases of these low excretors. What does that mean? Well, if you remember my first slide, I talked about an elevation. When you think about an elevation, you think about, if you think about your cholesterol level, and I've got a cholesterol of 200, and somebody who's at high risk of heart disease has a value of 6, 8, 900. Well, the problem is that these individuals have a level of 200, but they're at the same risk of somebody with the much elevated level. So they can have very minor elevations in any of the metabolites that I talked about, and still potentially have the same phenotype at risk of the same consequences. You can get elevations of C5DC in other metabolic conditions. For example, if you're an MCAD carrier, you can have elevations in C5DC. That is actually sort of a lab phenomenon, probably rather than a real thing. And you can get elevated glutaric and 3-hydroxyglutaric by intestinal bacteria with severe ketosis. So when you're looking at these from a diagnostic standpoint, even if you see the metabolites, you've always got to determine what's going on in the patient. So the first couple of years we've been doing this since 2005. This is something I'd always heard about. This is something that was that we were supposed to be aware of, but we hadn't seen any of these cases. When you haven't seen it, you don't really know what you're looking for. When we get deficiency samples, the amount of glutaric and 3-hydroxyglutaric was so high it would be hard to miss. At least I think I always got it right. And so what I'm going to talk to you about is initially 2 patients who were detected by expanded newborn screening that we determined are low excretors of GA1. And I'm not lying about this, they were detected in the same week. So if you guys do this and you know you get false positives for this, what are the chances your first 2 low excretor GA1s would show up the same week? If it was over a holiday, it would be the trifecta, right? And just to cut to the chase, both of these patients are currently treated with diet, and so far neither one has had an event. So we're hoping that the diet's helping with that. So how did we get started in identifying these patients? Well, the first thing that happened was the newborn screen. And here's the cutoff in South Carolina, 0.32. This is a gluteral carnitine on the blood spot. And you can see this value was pretty high, so we were pretty concerned about that. And the other thing that I neglected to mention before, for those of you that don't do this quite often, is both of these conditions, because they can be missed and because the abnormalities can be so subtle, South Carolina gets these patients after an initial screen. So if you have an initial screen for C5DC, we don't do a second screen, well, they may do one, but they're really going to key on the initial screen and what it was. And that's for both VLCAD and for GA1. And South Carolina, our institution, has even gone so far as we basically do follow-up sequencing for both of these disorders, as well as the metabolic test. We try to turn that around in about 10 days. So the first patient had a positive newborn screen. Again, the same week. You get these. So if you get these the same week, this is the one you're concerned about, not this little guy.
Yeah.
Just fault of human nature, I guess. So, and then when we did the plasma 0.54, we're like, okay, well, this isn't grossly elevated, but this is probably consistent with a low excretor phenotype. And so we got ready to send off the gene sequencing and found 2 mutations, one of which was consistent with a low excretor. So at the same time, we're working against this background, and we get 0.35, and we get 0.18 in the plasma. And I wasn't super concerned about this, but again, we weren't getting anything that was normalizing. We weren't getting any normal values. So my postdoc I had at the time, who's named Laura Pollard, and I'll show you a picture of Laura at the end, had read this paper from Mayo, where they looked at elevated urine gluteral carnitine in low excretors with GA1. And so she comes up to me and she says, hey, I want to try a urine azocarnitine analysis. And so I had grown up with I'd been trained with the chapter that David Millington wrote, that there's a specific line in there that says urine azocarnitines have limited usefulness. And so I was like, typical of a mentor, I apologize for any mentors in the group, I was like, yeah, sure, go ahead, do that, but do what I tell you to do first. And she comes back with this, that the urine gluteral carnitine is very elevated in this patient. Keep in mind that this one was normal, and so now we had this patient that was probably a low excretor, and this patient that we still weren't real sure about. And to the physician's credit, they didn't quit, and they sent off the gene sequencing on patient number 2 and found 2 mutations, one of which was novel, not associated with what was considered probably a low excretor phenotype. So seeing this value and this value got me really worried. There's a familiar reason why I'm losing my hair, but this doesn't help. And I get very concerned about this because I wonder, wow, how many 0.8 0.18 plasmas have I said were normal, especially, again, when I looked at the organic acids, it felt like they were normal. So, again, very good postdoc. Let me show you the data first. So what we did is we collected multiple urine and plasma samples from these two patients, and then a patient we had with glutaric aciduria type 2. And it's hard to represent this all on one graph because you're dealing with two different tissue types, so let me try to explain. And if I don't do it right, then you can send me an email and say, hey, you need to change your display format, okay? But basically what this is 1 is the upper limit of normal, and we divided all the values by the upper limit. Yeah. And so anything under 1 is a normal value, and anything above 1 is an elevated value. And this is the degree of elevation relative to the upper cutoff. Can we go through it again? No, we're good? Okay. So what you can see is that for patient 1, I feel like the urine glucocarnitine, which is the line in red, may actually be more persistently elevated than the plasma value. The plasma value tracks right around our cutoff. You could argue, hey, well, it's a problem with your cutoff drug. It's not a problem with the assay. But this cutoff is about 0.13. I mean, it's in line with what you see in the published literature. So this patient suggested maybe that the urine is a more consistent elevation and would therefore be a good follow-up test to identify your low excretors. Patient number 2, again, had this value, which concerns me to this day, which was right about the cutoff, but then showed persistent elevations. And again, the plasma went up here, but then sort of tracked again more like this. So when I look at these two graphs, the urine looks a little more consistently elevated than the plasma. It's not perfect. It's not without fault, but we're not going to get that with this disorder. At least that's my opinion. And then we have a glutaric aciduria type 2 patient, and you can see her values trend the same way. So, again, I wake up on a Friday night at 2 in the morning, and I'm worried, how many of these cases have I missed? What we need to do so. Now I'm like, oh, it's a great idea. Did I ever tell you I read that article? Because it was all my idea, right? Being a mentor, I told my postdoc it was all my idea. And so she was already way ahead of me. And so she pulled all of our plasma isocarnitine analyses that had mild elevations in C5 that we had urine sample on. And she ran all those for urine C5DC. And they were all normal. And I got a good night's sleep. And then she comes in my office and she says, well, now I went and pulled all the patients who'd been referred for a positive newborn screen for C5DC, but that we said were normal, because again, the urine organics can be normal in these patients, at least based on looking for glutaric and 3-hydroxyglutaric. And she said, but there's this one case. And the kid was normal, so we pulled that child back in, and one additional patient, I'll show you the level in just a minute. Plasma was mildly elevated, again, normal urine organics. At the time, we were also sending off for quantitative serum measurements for glutaric and 3-hydroxyglutaric, and those were plus minus on all these cases. Sometimes it would be normal, sometimes it wasn't. And you always got the report back, and the report says maybe normal in low excretors. So my friends in Colorado were not helping me out very much. And so we actually had the lab in Colorado do the sequencing, and they detected this mutation. And then we thought, okay, it's a carrier. But we couldn't get ourselves away from that. And so then we went and did the enzyme testing. And the enzyme testing was considered to be deficient by the guy who discovered the condition. And they said they've got about 3 of these low excretors who there's only one mutation, have a low excretor phenotype, and the enzyme is low. So where that other change is, what's going on, I'm not real sure. But this is just hard. I mean, and so what we've tried to do is come up with a way to minimize our send-outs and to get as much information from each positive newborn screen within about 2 weeks, so we can decide whether or not to go on and do what's considered the gold standard, which would be the enzyme test. And so this is where we were about 18 months ago when my postdoc, Laura, went on from her biochem fellowship to her molecular fellowship. And of course, I got my hands right in that, and I said, hey, why don't you do the sequencing for GCDH, and we'll start sequencing our patients rather than sending them out. And we'll just take every patient that comes in, we'll sequence the gene, and we'll see what we find. And we'll see how many of these kids might be out there. And so that's the sort of prospective study that we did. Whoops, I should this first. So here are the 3 patients. Here are the newborn screening values, 0.35, 0.55. I'm used to seeing values that are 3 in a good, I feel, in a situation that I feel good about the diagnosis. I like this value to be 3-4 times the cutoff. Not, I don't know what percent that is, 10%? And again, our QC values for CDC and other places agree pretty well. I mean, I think if you guys ran this, you'd be within the standard error of what we got. Here are the plasma measurements. Again, this is very scary because this value even went down on the second patient when we did it. But the urine gluteral carnitine was up. And when we've done successive measurements for gluteral carnitine, it stayed up around the 2 to 3 range. Now I can tell you we found one carrier, and her elevations are intermittent. She will every once in a while have an elevation, and then sometimes she won't. So I still don't think this is the greatest test in the world. But I feel better about it than some of these plasma values. So this is our prospective study on 19 positive screens for GA1 that we've done. This is the urine C5DC with 1.5 being the cutoff. And you can see all of those were normal except for a confirmed GA2 that we picked up. And I showed you before that GA2 will also have abnormal levels of gluteral carnitine. Here's the plasma C5DC levels. You can see we got 1, 2, what would be sort of mild elevations. And then over here, I just put interpretation, but essentially this is what the molecular findings were on all these patients. We sequenced all the exons. Did not do dosage studies, which I know you guys do. I'd be very interested to know if anybody's found any deletions, exomic, whole gene deletions of this gene, because that's what right now we're going to miss. And it'd be interesting even to take one of these 2 patients and see if they might have something like that. But some of these values, this 0.16 and 0.24, in my opinion, and maybe I just have a dirty machine, is almost just background. You've got to get up higher than this. This 0.45 is more real. And those of you that do this, and I didn't mention this before, know that there's crosstalk between C5DC and a couple of other acylcarnitines. So even though I'm calling it gluteral carnitine, there's actually a couple of isobaric compounds, or one isobaric compound that comes out at the same time. So I don't have an answer for you for GA1, and that's one of the reasons I'm here. But I'll tell you what we're going to continue to do is when the sample comes in, we'll measure plasma C5DC, we'll measure urine C5DC, and we'll try to turn around the sequencing within 2 weeks. Based on all that data, we'll go back to the family. Hopefully within 2 weeks, there won't have been any episodes, and we can give them a more accurate risk than just using the plasma C5DC alone. And I'd be really interested if you guys or anybody in the room or out there does serum butyric or serum 3-hydroxybutyric, and would be interested in taking a look at these patients, because I think that would be really interesting to look at another secondary marker. So when I was putting this together, and the reason I chose these 2 conditions is we started doing VLCAD sequencing about the same time we did GCDH sequencing. And so I wanted to sort of compare and contrast these 2 conditions because they are both sort of first tier, or as soon as the screen's abnormal, we got to get the patient in kind of disorders. They're also both widely known that you can have normal values on secondary testing, secondary biochem testing. So the incidence of VLCAD is much higher in the rest of the world than it is in South Carolina. or maybe we're just not picking them up. I'm not sure which is the case. But it's funny to me that the incidence of VLCAD is much higher, yet we have 3 times the number of VLCADs, and 3 times the number of GA1s than we do VLCAD. So something in those numbers is different. And with all due respect, while our football teams may be totally different, and everybody wants to separate it, genetically, we're not far apart. So if we're getting this data, I'd like to know what you guys are getting for VLCAD, because it seems like it should be more of a continuum. Same way with the data from North Carolina. So again, this is what keeps me up at night. Are there others? You got extra Ambien, send it to me. I seem to have a lot of those. So again, a lot of you guys may know this. VLCAD is it's an enzyme that's involved in mitochondrial beta oxidation. It works on basically acyl-CoA molecules that have greater than 12 carbons. You guys have people here that know a lot more about the specifics of this disorder than I do, so I'll shut up at that point. But the clinical features are, like a lot of fatty acid oxidation disorders, hypoketotic, hypoglycemia, seizures, kind of heart problems, and liver problems. And then, of course, like all these disorders, you can have a milder, potentially later onset disorder. For the lab people, the characteristic thing we're looking at is this guy, who's named C14-1 and C16. These are the things that are going to kick off our newborn screening. And I'll tell you, one of the things South Carolina is working on, so we're working on doing a much better job of getting ratios and what ratios to use and getting that involved in our newborn screening. Getting those ratios, if not reported, at least reported to the physicians. And we just had a meeting 2 weeks ago about this. And also, how are those ratios going to change in South Carolina in 3 weeks? Goes to the underivatized kit versus the derivatized kit. There's a lot of interesting stuff going on. And GA1 was one of the biggest concerns. And I can share with you guys some of the data, some of the preliminary data from our state lab. You guys may have already done it, and if so, I'd be interested to know what your results are. So this gene is located on chromosome 17. It's composed of 20 exons. And approximately 90% of patients with VLCAD can be picked up. Again, you guys do large deletion duplications, and I'm dying to hear if you guys have picked any up for VLCAD, because I'd be really interested to know, because those are the cases I'm going to miss with just standard sequencing. So we started this VLCAD gene sequencing in sort of 2008, 2009, and we're taking the same idea with our GA1 patients that we're taking with taking the GA1 information and putting it on the VLCADs, and that basically any positive screen receives full sequencing as well as the biochemical studies, but nobody, at least our physicians, don't really trust the biochemical studies, and that hurts me to say. But that's the way it is. And so, interestingly, and this is again sort of what got this talk going, is that we have approximately 20 positive screens since we started doing this that we've done this analysis on, which is about the same number we have for GA1. So you can sort of compare and contrast the two disorders. And so these are some of the results. I put the data from the affected on the top so you can sort of see what an affected individual looks like if you're not familiar with looking at. The cutoff I use for C14-1, C12-1, I think is about 3. Is that what you guys use? 2.5, 3? No? Wrong on that? And of course, there are other ratios you guys may use. C14-1, C16, C14-1, C2 is, I think, another good one. I'm sort of old school. This is the one I'm familiar with. This is the data from Duke. And so in these approximately 20 individuals, we have picked up 10 carriers and 10 where we couldn't find a mutation. And we took 5 of those 10 carriers and we had enzyme activity measured. And they were all, I'll show you that data in a minute. So one of the first striking things to me about this is, why are we picking up all these VLCAD carriers? And is it just because birth is a traumatic process and it's stressful and it's just pushing the pathway a little bit and you get the microelevation? If you guys have other ideas, I'd be really interested, but I'll buy that. And so, just to sort of go through this data a little bit with you, and this isn't really that exciting, but if you look at the C14-1 levels in normals and carriers compared to the affected, normal and carrier compared to the affected, I like this data but we're not where we were with GA1 of one we could potentially miss. We don't have one. Our patients are pretty classic. That's what I'm concerned about. So I'd be interested in knowing the data that you guys have on patients that maybe have a mild. So if you look at the enzyme activity, and this was all done in fibroblasts, I know there's some discussion about having a lab potentially here at Emory that would actually do this analysis in leukocytes. I think that would be a nationally utilized resource. It would be a great thing for a postdoc or somebody like that to learn because it's really needed for this disorder. So you don't have to have scared parents like me seeing their kids biopsy. And so you can see, I think the enzyme activity is pretty good at making sure that the patients aren't affected. All of our patients who were identified as carriers, none of them seem to have some mysterious second mutation that we could find. And that makes you feel a little good about doing the sequencing and sort of trusting it. And again, you guys are probably very familiar with this. I'm just getting into this. But there's one of the regional collaboratives, which again, Ronnie is leading our regional collaborative. But the Region 4 collaborative has the Mayo Clinic in it, and a very bright guy who a lot of you probably know named Piero Ronaldo. And I honestly believe, and I can say this with a straight face, that the data that they're putting together as a collaborative project is probably one of the most significant things to happen to newborn screening since we did the expanding. If you guys aren't familiar with this, basically what they're doing is they're creating a database of all the positive screens. What were the levels? What were the ratios? And then they let you see the data. And they put it in these complicated, once you learn the lingo, very interesting percentiles, whether the patient's affected. They give you target ranges for your data, how well is your state doing. It's impressive to me that all the labs are willing to share their data. They're willing to examine what they're doing and learn from it. I know in South Carolina, we spent an hour and a half the other day going through each of the cutoffs and where we were on this scale and what things could be moved, what ratios could be put in. And we're gonna save ourselves some money and save ourselves some time by decreasing our false positives. That's a whole nother discussion about how this thing works, and I'm not an expert at it. But what I wanted to show you is we took this relatively new tool. We took our 20 cases where we thought we had a good idea whether they were affected carriers or normal, and we put it through this tool to see what we might find. And so I hope I don't get in trouble for presenting this, but I am part of the Region 4 collaborative, so I can show you the data. This is the affected individual. These are the carriers, and these are the normals. So I would argue that this, based on our 20 samples, which is relatively small, we're a small state, it's the best I can do, seems to be a good tool to utilize for determining whether the patient has VLCAD or doesn't. I'm really interested, and this is the kind of thing that I think Piero and the group from Mayo, they've probably already got 10 people pulling all this data from all over. My 10 samples aren't that interesting to him, but I'd be interested at a regional level if we were to do this for all of our VLCAD screens and actually pull that data together and either present it to him, and if you guys are already doing it, you can have ours. Okay. When Brian and Ronnie asked me to do this, I'm not directly involved in newborn screening, and I'm not in one of the states that is really on the cutting edge. And so I thought the way to present this was to show you some of our cases and ask you guys to give me feedback on how we did and what we can share together. My feeling on where this is going, and this is what you have to sit through for your free pizza, so I'll make it brief, is both GA1 and VLCAD remain bad actors for expanded newborn screening, and they're not going to go away. It's just the nature of the disorder. I don't think we need to be overly concerned that we're missing all of these cases. At least that's my feeling. I think we're always going to be cognizant that that possibility is out there and be willing to go potentially that extra mile. And I really believe that further testing, finding additional tests, whether it's urine C5DC, whatever test your state can work into its workflow. Again, you guys have the opportunity to do second-tier testing because your labs are close together. I'm 70 miles away, and it's hard for me to do quick second-tier testing. But I can get the patients pretty fast, and I can come up with this kind of second-tier test to help me. And so the idea of what second-tier test you're going to do and how's it going to work. And finally, finding some bioinformatic tool that can help us minimize the number of false positives, reduce the stress on patients, but identify the kids who really need the treatment and really need the fast care that we all hope to provide. I think molecular genetics is just going to be a part of this, and I get in trouble quite a bit at the meetings I go to with our molecular lab when they ask me what the turnaround time should be, and I say, well, 5, and I say 5 weeks, and I say, 5 days. Molecular people in the group are already frowning at me. I understand. I'm bully. They don't like it when I come around. But I am certified in biochemical genetics. I do know how long it takes to do a PCR. I know how fast these capillaries work. And so I really think if you have enough people to do it, that you can really push this through pretty fast. Don't talk too much. Can't even look at you. So, I thank you for coming. Thank you for your time. Very interested in your thoughts. I didn't put you to sleep or give you any indigestion. The people that do a wonderful job in the DHEC lab as part of both the measurement and the follow-up. And it amazes me still that all the newborn screening that we do in different states is essentially done by people that would fit in probably this room. Or maybe this room and another half. In South Carolina, we can all get around the same table. And 60,000 samples with a 3-day turnaround time, or whatever it is just an amazing thing. The GGC metabolic team is this group here. It's made up of physicians, dietitians, some care coordinators, lab people like me. And then if you like the way the data work, these are the two women that put most of it together. This is Laura Pollard. If you know Lori, she's going to have, you can congratulate her, she's going to have her first baby. She's due a week from today, a week from Sunday. So she probably won't be in my next slide, or at least her baby will be in the next slide. And this is Irene DiBiase, who's on you. This is our sort of molecular biochem fellow and our straight biochem fellow. If you guys have any questions, I'll be happy.
We have a question from a participant on Unity. This comes from Hans Anderson from Louisiana.
Speak up.
What about the VLCAD pre-positive? Pardon me. What about the VLCAD pre-positive patient with 1 mutation and 2 polymorphisms?
So you get into the idea of polymorphisms. I would ask Hans, are these known polymorphisms or are these variants of unknown significance? So I'll try to answer both questions because Hans is like 5 miles away. What I would do and what we've done is we try to, if we can, get the parents on the initial blood draw so we have the parents if we need them. And I think that's one of the cases where you have to think about doing a biopsy and getting the enzyme done until we have the bioinformatics data to know whether they're really polymorphisms or not. But I think as we study this gene more, when we pool our data, we're going to find, you're always going to find a polymorphism. But as we know more of them, we'll get less stressed. What do you think? Yes? Okay, Hans, the group here says yes. Anybody else? Yeah.
So I'm just curious if you guys are using those Region 4 tools in the follow-up process routinely and at what stage you are?
Well, I have one physician, Nina, who loves to use it and does it on all of her patients. I have another physician who's somewhere in this graph who is not so interested in, just isn't as comfortable with computers. And so I'm a huch advocate of it. And it just makes it. The other thing about being a small state is we got our first 3-methylgluconic aciduria patient. Okay, I'm going to see one this century. I'm going to see one next century, or whatever it is. And I could look on that data and I could say, look at that, smack dab in the middle of the effective range. it was just an invaluable tool to me for these rare cases. I mean, if you're a big lab like you guys, or you're like New York or Texas, you're going to see these rare cases and you're going to know what they are. For somebody like me, it's really helpful. How good the calculators are, I think you've got to do stuff like this, and you guys are going to have to tell them what works and what doesn't.
Absolutely. You correct.
No, I'm not going to do that, but I'm just going to add a couple of comments. So Puro has added regional data now. So if you want to go on the website and want to look by region, we can have that access in terms of what the data provides. Also, the GeneDx have done a study, and they did find 13 to 14%, particularly with the fatty acid oxidation disorders, which had del-2 dips. So the percentage was much higher than I had anticipated. We started calculating in our clinic, and I don't have those numbers, but that data was presented at the ACMT meeting last year.
Why is that higher than you expected?
Because we never had seen that number, and in our patients we were getting 95% of the patients were coming up with some mutations. We didn't have large enough a population to look at, but now that as we are gathering numbers, we are falling within that range.
All genes have deletions in about 20%. So your a priori expectation
Would be
with no data is about 20%.
It was lower than 20%, though. It was
Well, 15% of a small number is the same as 20%. So it depends a little bit on the size of the gene, but you should expect 10, 20, 25% of the mutations will be a deletion or mutation.
So it confirms, but the data was quite large, though. The GeneDx data was fairly large, what they showed the sample size beyond our clinic. It was like hundreds of patients they had done. The newborn screening. But it was higher with the fatty acid oxidation, particularly with VLCAD. I remember asking that question, I can't recall, but that may be more closer to 18% or 20% in the fatty acid. So to answer your question, we haven't looked at, there are few we have gotten in our patients, but it's still a very small sample size.
But Tim, I'll put you on the spot. Why are you doing del-2 analysis?
Could you please repeat the question over the microphones to the people in the audience?
Okay, David is asking why you're not doing del-2 analysis.
Because I got my postdoc doing a lot of different things. But you're right, it should really be standard of care to do del-2 studies.
And it's available several places.
Right.
So our molecular genetics lab actually feels that we should not be screening the genes before we get the enzyme analysis done. So, I was curious to hear your
Right.
comments.
So the idea is that the molecular people say you should do enzyme first, and the enzyme people tell you should do the molecular first. And I'll honestly tell you that's happened to one of these cases where we sent off the molecular, found one mutation, and of course the bottom of it said, further enzyme analysis is necessary to determine the significance of these findings. So they do the biopsy, they send the enzyme off, they get the result back, it says, decreased enzyme activity. Molecular analysis should be performed to determine the significance of these findings. At some point you just got to say, it just looks like a carrier, and hope that you're right.
This is really an example where you have to take all of the data together. You have to take biochemistry, you have to take the molecular, and sometimes one or the other will give you a nice complete picture. If you do the molecular and you find two nonsense changes, yeah, that's what that is, and you don't need to go through biopsy and then. But if you have, especially with some of these where there's this abnormal newborn screen, the level of suspicion is not very high to begin with, you can start with gene sequencing. But if it's negative, are you going to go with which, if you don't have a high clinical suspicion to begin with, chances are it is going to be negative. And I think that's the specific case we were talking about, which to do first. Well, if this is it, you're pretty sure this is it. Yeah, molecular may give you a complete picture. But it really is case dependent, what information you already have, and then in a more complicated case, or a non-classification, you may need all of these pieces together to really figure out what's going on.
Right. So for anybody who's outside I'll summarize in Laura just said. For difficult cases, you need to sort of do all the analyses and look at the data and see what you can find. And I completely agree with that, and I think this little bit of data that I showed you supports that for both of those conditions. There was something else that she said that I was going to try to comment on. I guess I need my pizza. This is a great discussion. This is what I was hoping we'd get at. And if anything, it's just people sort of venting their frustration with these two conditions.
Any more questions? If not, I'd like to thank Tim again for coming today. Thank each of you for making the talk so successful. Be on the lookout. This is something that we're hoping to do fairly frequently, on the order of every couple of months. So I'd love to see you here in the next couple of months when we announce our next talk. So Chinese next time. Thanks again.
There's extra pizza if you want a piece on the way out.